So we are doing a video on PCR. For starters, what even is PCR? PCR stands for polymerase chain reaction. This process is basically a way to quickly amplify specific or target segments of DNA. Now, if you don't know what DNA is, I suggest viewing another video or reading a 6th grade science book. Using PCR, one can make millions of copies of a section of DNA in just a few hours. That's the same amount of time as the One Direction This Is Us video. Okay, so now that we've told you a little bit about PCR and what it can do, so what? What exactly is PCR used for? PCR can be used to trace human evolution, assist in DNA fingerprinting, detection of viruses, especially AIDS, and can help diagnose genetic disorders. But how does this process work? How do you go from DNA to research? Well, basically, it starts with a sample, like either a bone or a fingerprint, and then using an instrument such as cobas, one can extract that DNA. Then the DNA is put in the PCR machine, and the DNA is amplified and studied. So you're probably wondering what exactly happens in this PCR machine? Well, basically, it's a 3-step process repeated in at least 3 cycles. It needs ingredients, such as template DNA, DNA primers, nucleotides, and thermophilic polymerase. The 3 steps that each cycle has to go through include denaturing, annealing, and extension. The first step, denaturing, is basically heating the DNA at about 95 degrees Celsius. This separates the double-stranded DNA strands. The high temperature breaks the relatively weaker bonds between the nucleotides. The second step is annealing. This is cooling the DNA at about 55 to 65 degrees Celsius. Man-made synthetic DNA binds to or anneals to specific sequences on target DNA region. Primers bind to the beginning of the sequence that will be copied. It is cooled so that this binding can occur and that the DNA are ready to be copied. The final step is extension, which is heating the DNA at about 72 degrees, which allows the DNA primers to begin at the DNA polymerase to begin at the primers and create a new strand complementary to the template strand, creating more DNA. So we're going to take you through this whole process, but in order to demonstrate this process, we decided to act it out. In our presentation, the pool noodles will represent DNA strands, the pipe cleaners represent nucleotides, the purple cloth represents primers, and the yellow cloth represents DNA polymerase. The first step of each cycle begins with denaturing. The double helix is separated with the heat that breaks the weaker hydrogen bonds, creating 2 new single-stranded DNA. The next step in the cycle is annealing. In annealing, the primers bind to their complementary sequences in the DNA strand. The last step of the cycle is extension, where DNA polymerase extends the primers, adding nucleotides onto the primer in a sequential manner, using the target DNA as a template. The end result is 2 additional DNA strands, which have part of the target DNA included. This process is repeated in cycle 2, except at the end of the 3 steps for cycle 2, the total number of single-strand DNA with target DNA equals 8. The process is repeated again for cycle 3, except at the end of this cycle, there will be 16 individual strands of DNA, but only 2 strands will contain only target DNA and nothing else. That is why it takes at least 3 cycles to amplify DNA, because it takes 3 cycles to get specifically just target DNA replicated. As more and more cycles occur, more and more DNA can be made.